Good afternoon. It's a pleasure to have you here today at the webinar for the open call for tender on role of water used in the growing, handling, and processing of fruits, vegetables, and herbs on the spread of antimicrobial resistance. The reference of this call for tender is OC EFSA BIOHAW 2023-01. I am Giorgia Ciani from the EFSA Finance Unit, and I'm here today with my colleagues Beatrix Guerra and Ruben Barcia from the Biological Hazards and Animal Health and Welfare Unit of EFSA. My colleagues will shortly present you this opportunity for scientific cooperation with EFSA. Objective of this webinar is to present to potential tenderers an overview of the call for tender, which was published on e-tendering and on the official journal on the 22nd of March of this year, May of this year. This webinar will focus primarily on the scientific content of the tender. In fact, after my opening remarks, I will give the floor to my colleagues Beatrix and Ruben, who will guide you through the background of the call, the objectives, the tasks and deliverables linked to them, and the main selection and award criteria. I will conclude the webinar with few final remarks, and we expect that this webinar will not last more than 45 minutes. Before we start, I would like to share with you some quick housekeeping rules. So first of all, you will be automatically connected to an audio broadcast, and it's going to be a one-way communication. This means that you will be participating in listening mode only. However, during the webinar, you are given the opportunity to raise questions via a dedicated Q&A chat. You should see the Q&A chat box appearing on your screen right now. And you can use the Q&A chat box to type your questions. As this webinar is held in English, we would appreciate to receive also your questions in English. After the webinar, your questions will be collected, anonymized, and answered publicly on e-tendering in the question and answer section. You can use e-tendering platform also later after the webinar to raise any question you might have. The webinar will be recorded, and the recording will be made available on EFSA website and on e-tendering portal of this call for tender short after the webinar has taken place, together with all the other tender documents that are already available there under the library section. After the event, participants will receive a link to a survey to evaluate the event, and we would very much appreciate to receive your feedback, which is very important for us to improve any future events of this kind. Now, I have finished my introductory part, and I will leave the floor to my colleagues Beatrix and Ruben, who will drive you through the main features and content of this call for tender. Beatrix is the first speaker, so Beatrix, the floor is yours. Thanks.
Sorry, Georgia, thank you very much. And in fact, what we intend with this presentation today is to let you through the technical specifications in a general way, also trying to clarify some issues that we consider relevant for you to know. And of course, you can find much more information in the technical specifications in a detailed way. Following this, we will present you some of the main aspects of the background, of the objectives and tasks, also from the deliverable and meetings, and what is important for you as a selection and award criteria. Following this, I would like to tell you why we came with this call. And there were two main reasons for this. The first one was that we published in 2021 a scientific opinion focusing on the role of the food-producing environments in the spread of antimicrobial resistance. And out of this scientific opinion came that the production of plant-based foods, so vegetables, was still an area where we have less information regarding antimicrobial resistance, and also that the water was an important source of in many of these food-producing environments that could be a source of a resistant bacteria and resistant genes. So it really came as that different waters could be an important source. We consider this, we dig into it in the one conference that in the session for antimicrobial resistance, and I included in this presentation that will be available for you, the link to it if you want to know more. The other reason to launch this call was that in parallel we have another tender that is ongoing, and in this tender we ask, the tenders are looking for the microbiological hazards associated with the use of water in the post-harvest handling and processing operations of fresh and frozen fruits, vegetables and herbs. In this call, they are looking at the microbial hazards, excluding AMR. That's why what we intend now is also to complement this. And we launched the call based on EFSA funding regulations and also considering the EFSA's 2023 draft work program for grants and operational procurements. Going more specifically to the topic that we want to cover, you know that we have expected changes on the water availability in many parts of the world, including Europe. And it's very clear that there is a risk of very limited water that could be available for irrigation and washing of food products without considering water reuse. So the European Commission is well aware of this and trying to look for solutions. They published in 2020 the legislation that we call Water Reuse Regulation that is on the minimum requirements for water reuse, and is really a regulation that tries to facilitate and encourage the practice of reuse of water to be used in agriculture. And in parallel, in the legislation on hygiene of foods, it's also possible, it's stated that it's possible to reuse water in the food processing environments. So in the food processing plants, the water can be reused as long as it does not imply any risk for contamination. So having this in mind, it's very clear that there is a need for water reuse. On the other side, we are aware that this reuse of water could be linked to the presence and possible potential spread of antimicrobial resistant bacteria and genes in these waters. And in fact, in these waters, we have found bacteria that can be considered of public health importance. So, on the other side, we also know that there are some studies, but in this area is the less known area. There are the number of papers focusing on Europe is still low and is now increasing. And one of the things that we have clear is that with monitoring and surveillance programs on AMR, we can generate really important data that can set the start for prioritizations in future actions. And to do this monitoring and to do this surveillance that is already implemented in other areas, we need still to have good methods for it. There is a lack of standardization. Not all methods are suitable to detect things that are in very low levels. For example, that can happen in these environments. This is an area that needs to be improved. Something else that we recognize as a gap is that we don't know so well which is the efficacy or the effect of commonly used water treatments during the reclamation and reconditioning treatments, how they affect resistant bacteria and resistance genes. And here it's important because on the one side, there will be for sure, if we diminish the number of bacteria, in general, also the number of resistant bacteria will be diminished, but we don't know if some of the treatments can really select for resistant bacteria that very often also have genes that allow them to resist disinfectants. Knowing all this and also knowing that the water reuse legislation indicates risk management measures that could be used to follow the quality and the monitoring with regards to reclaimed water. In this legislation now, it is included to monitor AMR among the additional requirements that could be considered. So we know the gaps. We have a legislation that allows us to look also for resistance, and we consider that it would be good to look now at this. How is the situation? Before we start, I would like to say that I'm talking all the time about bacteria of public health importance, and this can has really different meanings depending on the context that we are. You know that there have been several lists of prioritization of bacteria that are important with regards to antimicrobial resistance. And this context is different if we are looking at clinical, veterinary, food, if we are looking for the lack of antimicrobials against, for example, if we look at the WHO list. And within this context, the context of the current call, we are really looking at resistant bacteria and resistant genes of public health importance within the One Health approach. And considering that we are looking at a food producing environment. So you can get more information about this in what we call the EFSA AMR environment opinion. You also have the links in the specs. And in these areas, we know that there are many info gaps in these commodities, in the vegetables and in the waters. So we would like you to give priority to include at least those bacteria that we consider of highest priority for public health in food producing environments. This would mean to include Enterobacteriaceae such as Klebsiella, Salmonella, E. coli, that is the target in every monitoring system on AMR, similar to Enterococci. But we would like also to have some data on Acinetobacter, MRSA, Pseudomonas aeruginosa, and also included in several ongoing monitoring programs, extended spectrum beta-lactamases and carbapenemase producers. You can, of course, include any other bacteria that you would like to include. You need to justify why. You can also include isolates from other ongoing monitoring programs. I mentioned, for example, those collected through the water reuse regulations, or also from other research projects. Always that you fulfill the requirements that we describe in the different tasks, and or you can add them as additional isolates, but you need to have the permissions to use these isolates for the current project. Regarding the foods or vegetables, considering that we focus on fruits, vegetables, and herbs, I give the floor to my colleague Ruben.
Thank you, Beatriz. So regarding the vegetables, we show in this table some examples of different commodities that could be suitable to include in the study. But of course, you can select the commodities that you consider, always trying to represent the variety of fruits, vegetables, and herbs. To do so, you can take into account the general commodity categories described in the EFSA opinion of 2013 on pathogens in food of non-animal origin, and also the different crop types included in the legislation. Here, the first crop types that we see in the column of crop types are the one closer to the soil, so in direct contact with the water. But you can also include the above ground high growing crops if you consider it relevant. All the crop types are intended to be eaten raw, and depending on the crop types that you will select, together with the irrigation methods used, these two factors will determine the quality of the water needed as stated in the legislation. And with this information, I give the floor back to Beatriz.
So thank you, Ruben. So the overall objective of this call is really to close a direct contract with you so that we can gain insights on the occurrence and variety of the resistant bacteria and resistant determinants. I'm talking all the time about yeast or genes, but I really mean genes, plasmids, or total resistomes. So these include all. And we wanted to do it both in reclaim water and reuse processing water that can be used during the pre-harvest or post-harvest with regards to fruits, vegetables, and herbs. So we want you to focus on the water, but also on the foods themselves, so that you can really help us to assess the role of this water in the spread of AMR to these vegetables and in different European regions. We offer you a maximum budget of 950,000 euros to do the work, and it should last for 36 months from kickoff meeting. We have distributed this broad objective into 3 specific objectives. So the first one is really to try to optimize these methods that will be used to perform the studies so that they are really sensitive for those bacteria that could have a low charge. And Ruben will provide you more information about this in the next slides. The second objective, we want you to generate new data on the occurrence and potential spread of bacteria and these genes as a potential result of the use of the reclaimed water for irrigation. And in this case, we would like you to focus first on the urban waste treatment plants and reclamation facilities, taking samples there so that we can see something about the water treatments, but then to look at the water in the growing fields, and then to look in the same growing fields to look at the vegetables, because we want to follow the line. The objective 3 is the generation of the new data on the occurrence and potential spread of these bacterial genes as a potential result of the use of reused processing water. And here we want you to concentrate on the food processing plants. So to take samples there from the water and the foods. And here we make a differentiation is in those processing plants where they don't use any water treatment after being reused, after using the water, and those that really carry some water treatments and disinfection after having reused the water before using it once more. According to this, just for you to know that all objectives are interrelated, so the methods that you develop needs to be used in the other objectives, that you will look for treatments, and that you will look, as I said before, to follow the lines, what I have at the beginning, what I have at the end, what I have in the water, what I have in the foods. For the next time of the presentation, we will present you the tasks per objective, and following this, I begin with the task 1. This task, well, when you do the proposal, and this is what we will evaluate, you need to present us a clear study with regards to what you aim to focus on, to what you want to do. After our kickoff meeting, you will have the opportunity to refine the study if needed. And what we need to have from you is very clear which are the detection methods that you are going to test, the target bacteria and genes, the countries, having in your mind that we focus really on EU, EFTA countries. You need to provide us the number of samples, sampling points, sampling times, which are the specific compartments that you want to focus on, which are the commodities, the type of crops and foods, and then the water treatments, if you have water treatments or not, if they are chemical, physical, or combinations. So as I say, you need to present this in the proposals, considering these different variants, and then you can have the possibility to modify or adapt after our discussions or present more detailed information. Regarding the objective 1, I give back the floor to Ruben.
Thank you, Beatriz. The objective 1 is the one referred to the methods. These methods will be needed to perform the objectives 2 and 3. And the methods can be selected among the available ones or to be developed by you, but in any case, they must be optimized for the samples under consideration. The objectives need to be a combination of culture and molecular methods as described in these following tasks. The task 2 asks for the optimization of culture-dependent detection methods. The task 3 asks for the optimization of PCR-based detection methods. And the task 4 asks for the optimization of metagenomics-based detection methods to characterize the resistome of the samples to be analyzed. For all of the tasks, the detection is about the specific detection of antibiotic-resistant bacteria and antibiotic resistance genes of high priority for public health, selected among those mentioned by Beatriz before and included in the call. And also for all of the tasks, you will need to deliver protocols as well. And this is all the information for objective 1. I give the floor back to Beatriz.
Thank you. So for objective 2, as I told you before, we concentrate on the irrigation water. And in this case, we ask you in the task 5 to look what happens in the urban wastewater treatment plants and reclamation facilities. So here we are interested to know how the different treatments that happens in the plants affect what comes out in the effluents. And we ask you to include at least 6 processing plants, 6 treatment plants. At least 3 of them should provide the water to the fields in the next tasks. And they should be selected in 3 different countries, at least 3 countries, European, EFTA and EU. And we ask you to take samples at different sampling points. This could be the raw influent, after the tertiary influence, and the final influence. And at different time points, because we are also interested to know if there are some seasonal variations. In the task 6, we ask you to generate this data in the growing fields. So we want you to analyze the water that is used for irrigation, or also in these fields if this water is also used to wash the foods. So we ask you to include at least 6 fields. 3 of them at least should have the water coming from the previous water treatment plants. And from these fields, you will analyze in this task the water, in the next task the food. We also ask you to take samples at different sampling points, very close to the irrigation, so very close to the points where the water will come into contact with the foods. And as I said before, if you have some tanks to wash the fruits, you can also take the samples there. And we also ask you in different time points. This means different days along the year or during the production of the foods. We also ask you to collect in these fields the foods that were in contact with the water. So you need to include at least 3 different crop types from those similar to those described by Ruben before. And as I said before, this is 6 fields needs to get the water from different water reuse systems. You can add other fields that have the same water, or you can also test different commodities that are in these fields that they have the same water, but they are different commodities. This can be added additionally. And once more, you need to take the samples at different sampling points and at different time points. For objective 3, I give back the floor to Ruben.
Thank you, Beatriz. The objective 3 is about the use of reuse processing water in post-harvest, and it is split in 4 sub-objectives to cover different aspects. The first sub-objective, the task 8, is about the generation of data on the occurrence of antibiotic-resistant bacteria and antibiotic-resistant genes in the reused processing water used in handling and processing operations of fruits, vegetables, and herbs when the water has not been subjected to any disinfection treatments between the uses. So accordingly, the samples need to be reused and treated water coming from at least 6 different processing plants, ideally of different sizes, from at least 3 EFTA countries, and producing different commodities and different types of foods that will be analyzed in the next task, in task 9. So the water samples, thank you. The water samples need to be collected at different time points in the same day or different days, and need to be collected at different sampling points during the processing operations. For example, before washing and after washing. These water samples will be analyzed with the methods agreed in objective 1 as for all of the tasks, and also it will be necessary to collect information on the characterization of the processing water. Similarly, the task 9 also asks for the occurrence of ARBs, antibiotic-resistant bacteria, and antibiotic-resistance genes, but in this case, in the fruits, vegetables, and herbs during and after the processing operations in processing plants that use reused water that is not subjective to disinfection treatments. So the processing plants need to be the same from which the water samples were taken in task 8, and you have to represent at least 2 food commodities selected among those used in task 7, and also at least 3 types of foods, fresh, fresh cut, or frozen. So for example, if as food commodities you chose lettuce and strawberry, for the types of foods, the lettuce can be found processed as fresh or fresh cut as ready to eat, and the strawberry can be found processed as fresh or frozen. These food samples need to be collected at different sampling points during the processing operations again, and also ideally at different times along the day or at different dates. The task 10 is the same as task 8, so once again we're talking about water samples, but here the reuse processing water has been treated between uses. So So the idea is to assess the effect of as many disinfection treatments as possible on the spread of antimicrobial resistance. So correspondingly, the task 11 will be the same as task 9. So we're talking about food samples, but in processing plants that use reused water subjective to different water disinfection treatments. So in this case, the processing plants will need to be the same from which the water samples were taken in the previous task. And with this, I give the floor back to Beatriz.
Thank you. Well, once more, as it happens with task 1, we have here a task that is supra-objectives, and it's the task 12 that is really the integration of the results of the studies performed before for objective 2 and 3, comparing these results with those from the published in the literature. So what we want is really to see if you were able to identify potential links between the occurrence of these bacteria and genes in the reclaimed water in the treatment plants, then what arrives to the fields and is used in the fields, and what you find in the foods in those fields. Here is very important, and we ask you both here and in the next objective also to look for all the additional information that you can get that could be relevant to explain the results you find. For example, if in the fields there is the use of organic fertilizers, if these fields are open to wild animals and many other risks that could be also potential links for resistance spread. We also want to see not only the links with the reclaimed water, but and irrigation. We want to see also what happens in the processing water and in what you find in after the different operations that Ruben explained you. And we also want to see if there were some differences with regards to the different water treatments, use or not use of water treatment. And in this case, perhaps you can say us really which is the most efficacy treatment that should be applied or is feasible to be applied. And we want you to give you us some information about the results with regards to differences regarding the different commodities, type of crops, irrigation methods, European regions, as you can find, considering there is heterogeneity of the samples tested or not. So this is really what we want you to explore. We have set some meetings that we would like to have with you, with the consortium that will obtain this procurement, and the meetings we really intend to have one physical meeting with you at least once a year, apart from the kickoff. We wrote in the technical specifications hybrid meetings, but really we want you to be in place, and only if something really unusual happens after the COVID, we are prepared for everything, then you would not be there, but we really intend to have at least one person from each person of the consortium there. So we have asked you for some deliverables. The first one is really this fine-tuning of the study design to set really to describe very properly the experimental setup and the planning. Then we ask you for a first interim report asking for the results and protocols needed for the methods, detection methods. And in general, we ask for these deliverables always 1 month in advance of the meeting, so that we can have time to read and give you some feedback on the reports, so that we can discuss them thoroughly in the meetings. We decided not to ask you to do some task in 1 year and then other task in another year. We let you freedom to perform objectives 2 and 3 as you want in parallel or one after the other. But we want to see in these interim reports really an update of all the activities that have been done, to see if there is any need to intervene at any point, to see if something is not going well. So that's why we ask you for all these interim reports. And in the final report, you need to include everything you have done. And we will have some ping pong from you after the final report is approved. You have seen also in the technical specifications, and I will go now very general on them, that we drafted the selection criteria and the award criteria. So the selection criteria, we ask you for some requirements at the professional capacity is really for the institutions that are member of the consortium. And then we ask for specific expertise needed in the members of the consortium, in the specific persons. But in general, what we ask you is to have experience on antimicrobial resistance, water and food, onto water reuse, molecular detection, bioinformatics, and problem formulation, that is really writing reports, and project management. With regards to the consortium members, the experts, you need to have at least 4 experts that cover this expertise. One person can cover several. And this is what we are asking you. The person that will do the project manager will be the person that is in direct contact with us, representing the consortium. We also have some requirements regarding the technical capacity, and this is that you need to ensure that you have the infrastructure needed to take the samples, to analyze them, to draft these reports. You also need to have the permissions to access to these different establishments or compartments, no? that you can have access and to the processing plants, to the urine waste water treatment plants and reclamation facilities, and also to these growing fields. We ask you for some letters from them. You need to let them very clear that all data will be anonymized. Nothing will be published where they can be identified. This is not the object of the call. And you also need to have the library and digital collaboration platforms and needs that allow you to perform these studies. Regarding anti-English, that is not included here. With regard to the award criteria, we have 5 that relates to the quality, or what we call quality criteria. And here you will see, or I have already seen, that there are some of them that have more weight than others for us. With regards to this, we have the first one, the understanding of the assignment and the task required. So here what we want really to see is you have the knowledge and the state of the art on how the things in this area are developed, about production in Europe of these vegetables, about legislation, about methods suitable to be used, and this is something very important for us. Then, with regards to the methodology, we want you to propose step-by-step clarifications why you selected these things that you are proposing, with the justifications for them, and also the task distributions will be very important. This is with regards to the project management and organization of tasks. Here we want really to have a Gantt chart. If you offer, is distribute them among work packages, who is going to do what. This is what we ask you. With regards to the risk management, here we want you to present which are the risks that you identify at the potential mitigation measures that you want to offer. And then the measures to the quality of the deliverable. This has different topics. It can be the quality of the reports. It can be the quality of the data that you are obtaining. It can be the quality of the metagenomics or sequencing data. I don't know if you want to have any advisory or not. So this is what we want you to have a look. And for the two first points, you need to pass a threshold of 60%. And this is not needed for the others. But in total, you need to have from all the awards, 70%, 70 out of the 100 maximum points that you can get. And here there will be the awaiting regarding the price. So the price will be also taken into account to have the final selection. And you can see how this works in a formula that is presented at the end of the document. And with this, I would like to give back to Georgia the floor.
Thank you, Beatriz. So with this, we have reached the conclusion of the webinar. We can see questions raised in the Q&A chat box, and as explained earlier, all the questions will be collected, analyzed in detail, anonymized if needed, and answered publicly on the e-tendering portal in the question and answer section. Very important, you can raise questions, any question you might have also after this webinar in the dedicated section on the e-tendering portal. So please use the question and answer section on the e-tendering portal to submit any question you might have. You can click on question and answer section and click on create a question to submit your question to EFSA. As also stated at the very beginning of the webinar, this whole webinar has been recorded. The recording and the slides will be published on EFSA website and on the relevant e-tendering page of the call for tender, together with all other tendering documents. I would like to thank you very much, my colleagues Beatriz and Ruben, for presenting today this call for tender, and especially a big thanks to all those who participated today. Thanks for having taken the time to listen to us. Bye.